drugs and how they work. So pharmacogenomics combines these two disciplines to show us how our genetic profile or how our genes affect the way we metabolize drugs, or how our genes affect the way medicines work in our bodies. It also answers questions like, why does some drug work well for one person but fail completely in another? And why do some people need a lower or a higher dose of the same drug? So through these efforts, pharmacogenomics leads to personalized medicine. That is medicine or medication, or even like a treatment plan that is designed and developed just for you as an individual based on your genetic profile. And you can also take PGX as this. This is actually something I was thinking about a few minutes into the meeting. We want to buy shoes for all Africans. And then obviously we can't buy just one shoe size. Obviously it can't fit all of us. So what pharmacogenomics does in this picture is that it helps us to know what shoe size will fit which people. So it will help us to know the actual shoe size for the right person. So what are some of the problems we have in our current healthcare? Number one is high rates of adverse drug reactions, ADR for short. Now, adverse drug reaction is essentially any unwanted, unexpected, or harmful response to a medication when it is used at normal doses. We are not talking about overdose or underdose here. We are talking about drugs that are being taken at their normal standard doses. So an example is hypotension that is caused by a high blood pressure drug, let's say clopidogrel. So a patient reports to the hospital with high blood pressure, and the doctor prescribes clopidogrel, expecting that the drug will help to lower and manage the blood pressure of the patient. But this drug lowers the blood pressure way beyond the normal into the abnormal. That is the too low range, causing hypotension, and it's serious. It becomes very serious for the patient, because now the blood pressure is too low that the body is not able to carry enough blood to organs like the brain, and it can lead to unconsciousness or even collapsing of the patient. Another example can be like a stomach ache from taking an antibiotic, or even an organ damage but from taking a medicine. And the next problem is the trial and error prescribing. That is something that is going on currently, because we don't have pharmacogenomics in the picture of our healthcare. Normally, what doctors do is that a patient reports to the hospital with a specific disease, and the doctor would prescribe a drug that is known to cure that disease. And then the doctor would monitor the patient for some time. And if the drug is not working, the doctor will change to an alternative. And then it goes on until the doctor gets to a medicine that is able to actually cure that disease, sorry. And that leads to wastage of medicines. And wasted medicines means wasted money. We waste a lot of money by not incorporating pharmacogenomics into our healthcare. And there's also high cost of repeated treatments. Like I said, if the drug is not working, another drug. So it all requires a lot of money. A lot of money goes to waste. So why does pharmacogenomics matter in Africa? I mentioned Africa earlier. Why did I do that? Why do we need to pay more attention to pharmacogenomics in Africa? Number one, like I mentioned, Africa has the most genetic diverse population in the world, and that means higher variation in drug metabolism, right? And higher variation in drug metabolism also means that we have higher risk of ADRs, adverse drug reactions, and higher likelihood that global drug guidelines won't fit perfectly. That like a wrong shoe size for the wrong person. So from that, yes. So like I was saying, sorry for the interruption. Won't fit the global drug guidelines that are being developed won't fit perfectly because the companies, the pharmaceutical companies that produce these medicines for curing these diseases don't consider how rich our genetics is. So they bring generic medicines that are generally known to solve the issue without considering the actual genetics of our population. And then it causes us a lot of money. So many ADRs in these departments. We have other departments that also record cases of ADR, but these cardiology, mental health, and infectious disease, they record higher numbers of ADR. So these ADRs can be predictable if and only we understand a patient's metabolism. And then pharmacogenomics helps us to do that, helps us to understand and tailor treatment according to individual based genetics. So this is one of two scenarios that I have prepared to help us appreciate how much we need pharmacogenomics in our healthcare. Please, if I'm speaking too fast or if something is unclear, you can draw my attention. Thank you. So in this scenario, we have a child, a CYP2D6 ultra rapid metabolizer taking codeine. So this child undergoes tonsil removal surgery. Let's say the child um, was suffering from tonsillitis, and the doctor recommended that he goes to remove the tonsils. So after the surgery, after removing the tonsils, he is given codeine, which is an opioid for pain relief. But later that night of taking the medicine, starting to take the medicine, the boy is rushed back to the emergency room of the same hospital with breathing difficulties. And then after a few minutes, the child goes into coma. Now, what's really happened? Studies show that many African children are CYP2D6 ultra rapid metabolizer. And this boy was one of them, was like part of this population of ultra rapid metabolizers, which means that his body converted codeine into morphine too quickly. Codeine actually is a prodrug that needs to be converted into morphine. That is the active form for the therapeutic effect to be exerted, for the pain to be relieved. So the boy, because he is an ultra rapid metabolizer, his body converted the codeine that he took too quickly into morphine. So the morphine levels in his body shot up rapidly, and then that caused the breathing difficulty and then the coma that he went to. So how could have pharmacogenomics prevented this. If, let's say, if a pharmacogenomic test was done before the doctor prescribed the medicine to the boy, it would have shown that codeine is unsafe for the child, and then the doctor would have prescribed an alternative, which would have been safer. So what pharmacogenomics does is it doesn't just guide treatment, it prevents emergencies like this even before they happen. Okay, so this is the second scenario. And based on what we have learned so far, I would like someone to help us break this down. What do you think is happening, or what do you think will happen, or what happened? Anyone?
Please kindly simplify if you want to answer.
Pardon?
So I say they should signify if they want to answer.
Okay, okay. I think one person raised the hand. Yes, please, Pascal.
Okay. You can unmute your mic now.
Oh, from the image, I'm thinking that the enzyme, this individual probably doesn't. She bears a variant of the enzyme which doesn't induce metabolism of the clopidogrel. So the customary clopidogrel is used in management of the risk of heart attack. So since the person, the person's enzymes are not induced to metabolize the drug, or rather, since the person's enzymes are used to, since the person it doesn't activate it, it leads to a risk of second of heart attack. I mean, that's what I mean. Let me come again. Clopidogrel is used in the management of heart attack, and I feel like when the person takes it, the person is a variant of this enzyme, which breaks down the drug, or is that the case? I'm thinking. I don't know if clopidogrel is a pro drug, and then increases the risk of the heart attack because it doesn't work. It breaks it down too fast.
You said it breaks it down. The patient breaks the drug down too fast.
No, no, breaks the yeah. If the person's enzyme, if it's maybe it induces the metabolism of the clopidogrel too fast. That's if clopidogrel is not a pro drug. I don't know if it's a pro drug from this slide
It's
I'm
it's
seeing.
it's a prodrug. It's a prodrug.
Oh, okay, it's a pro drug. So if
It
it's a
has to
pro
be
drug,
activated. Yes.
yeah, if it's a product, that means it doesn't induce the metabolism quickly enough, and it doesn't lead to the active metabolites which does the work.
Yes, 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 that is correct. Thank you very much, Pascal. So what he said is the case here. So it is probable that this woman will return to the hospital with another heart attack, and everyone will be confused. And I'm sure some people would even relate it to a spiritual attack or something. But the answer is simple. The genes of the woman, she is a SIP2C19 poor metabolizer, or she has a variation in a SIP2C19 gene, the gene that is supposed to encode for the production of the SIP2C19 protein needed for the metabolism of the drug. She is a variant of that gene, which means the drug it's not able to exert the therapeutic effect. It is not broken down at all. So she can be swallowing, or she can be taking the drug every day, but the drug is actually never working in her body. And apart from the risk of a second attack, it can be affecting her organs. It can be accumulating drug toxicity in her body. So these kind of issues add up to how important pharmacogenomics is. So up until this point, you would agree with me that pharmacogenomics has the potential to transform health care systems, and especially in Africa, where hospitals often struggle with high patient loads, struggles with limited resources, and the heavy financial burden of ineffective treatments that are actually not working. So by understanding how a person's genes affect their response to medicines, hospitals and other health institutions and even health care practitioners can significantly reduce these avoidable risks and improve treatment outcomes and also save a lot of money. So one of the most important advantages of PGX in hospitals and HMOs is the reduction of ADRs. So many ADRs or adverse drug reactions happen not because the drug is bad. Sometimes the drug is good, or most of the times the drug is good, but the patient's body processes it too slowly or too quickly due to their genetic makeup. And then in Africa, where genetic diversity is extremely high, the risk of unpredictable reactions is even greater. But with PGX testing, hospitals can identify these risks before treatment even begins. And this will lead to fewer admissions caused by drug toxicity, fewer treatment failures due to medications that don't work for certain individuals, and fewer cases of patients reacting negatively to normal drug doses. And as obviously, as the drug reactions decrease and treatment outcomes, sorry, as the drug reactions decrease, the rate of drug reactions decrease, obviously the patient's outcomes are going to improve. So patients will start responding to the right treatment sooner, because with pharmacogenomics, the doctor or the healthcare practitioner would know the right medicine for the patient, the medicine that is actually going to work for that patient. And then their symptoms will stabilize faster and they will experience fewer side effects from the drugs that they take. So instead of following the traditional trial and error model of prescribing, where a patient must try multiple drugs before finding one that actually works for him or her, pharmacogenomics gives doctors a clearer roadmap from the start, right from the start. So they don't have to go through this trial and error or try different. Right from the start, the doctor is aware of what is going to work for the patient based on the genetics, which is an important factor at play here. And also patients feel better sooner, and then they will trust their treatment more. And then they are also likely to remain adherent. So when they experience any health complications, I'm trying to explain at the adherence here. When they experience these health complications, they would not stay in their houses and then go to the nearby pharmacy to get a drug to buy and treat themselves. They would go to the hospitals because they know that when they go, what the doctor is going to give them will actually work. So they build that trust and then become adherent. Also, better treatment outcomes will lead to shorter hospital stays and lower cost per patient. So patients receive the right medication from day one. There are fewer readmissions, patients returning back to the hospital, and then there are fewer prolonged admissions. And then the complications the level or the number of complications requiring expensive interventions, expensive drugs, and buying more drugs that are actually not going to work. These complications reduces. So for hospitals and again, Africa, we have a lot of hospitals that struggle with overcrowding, beds becoming unavailable way too quickly, and then resources being used um, more effectively. So pharmacogenomics helps in this regard by making the beds available more quickly, because the patients are not going to stay in the hospital for long. They would get treated very early and they would leave, and this will increase the number of beds to receive other people that are also suffering from other diseases. Another benefit that pharmacogenomics gives to hospitals and HMOs is evidence-based prescribing. So, like I said, in many settings, and especially in Africa, where pharmacogenomics is not heavily embedded in our healthcare, clinicians, healthcare practitioners, they heavily rely on guidelines and their own experience, but do not always have a personalized information to guide their clinical decisions for their patients. And pharmacogenomics fills this gap. So instead of them prescribing based on general data or population averages, doctors prescribe based on what will actually work for the specific patients they are dealing with. So precision medicine, also personalized medicine, increases the confidence in clinical decision making. Doctors would actually be confident when they are treating their patient, because they know that this method they are using or what they are giving their patients is working, is going to work. And for health maintenance organizations, evidence-based prescribing will reduce unnecessary claims, and then it will support sensible use of medicines and also improve the overall sustainability of healthcare facilities. So efficient workflow and then improved member satisfaction is also another great benefit that hospitals and health maintenance organizations derive from pharmacogenomics. When pharmacogenomics is integrated into hospital systems, especially in the electronic systems, it will allow the doctors and the other practitioners to receive an alert when a certain drug is not suitable for their patient. And this really improves the workflow of the the facilities, the workflow of the staff in charge of healthcare of the patient, and everyone is going to be happy, very happy. So we move on to integration. So integration, we don't need to begin with all drugs. Okay, we can start with a few drugs that are common and we know are most harmful, or they 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 fail in treatment. So some of these drug gene pairs are clopidogrel and CYP2C19, TB drug, isoniazid, NAT2 antidepressant, and CYP2D6 or CYP2D19, and then codeine and CYP2D6. And then we can also identify key departments. So this list is not exhaustive. We have other departments that also really need pharmacogenomics, but these departments record higher cases of situations like adverse drug effects, cardiology, oncology, psychiatry, and what have you. So after this, we can go on to choose a testing model. So we can either choose reactive or the preemptive option. So the reactive means you order a PGX test, or they perform a PGX test when it is needed. For example, before prescribing clopidogrel or before giving codeine to a child, and it will be useful in hospitals with lower budgets. And for the preemptive testing, it is actually the long-term approach. So you test the patient once, you get the data, and then you store the data in their health record. So like when you go to the hospital, you have your health record. This time your genomic data would be part, and then it would be embedded in the electronic system. So this data can be used for as long as the person lives. So let's say a man is, or a woman is 35 years old and gets PGX testing done. 20 years later, this person can or the doctor or the healthcare practitioner in charge of this person, when he or she gets hypertension or has to be put on a hypertension drug, this data from the pharmacogenomic testing can still be used even after like 20 years. And then I mentioned integrating PGX into electronic medical record systems. That is the EMR, electronic medical record systems. And I think this part is very important and is very innovative. It will actually it is needed for healthcare transformation, especially in Africa. So here's how it would work. When a doctor prescribes medication, the EMR would instantly check the patient's genetic profile and tell the physician whether the drug is safe or unsafe. So it can give a green indication, or there will be a green indicator telling the doctor that this drug is safe, or like an orange or a yellow indication signifying that the drug is not unsafe or is poses warning to the doctor, and then maybe a red alert to tell the physician that the drug should be avoided and it is likely to harm the patient. And then this information can be used to build local data systems, and it can serve as evidence for national guidelines, and like helping these companies, these drug producing companies, to know what and how to manufacture the drugs for a particular population. Okay, so I have a question for us all. You are a medical doctor, and let's say you're a medical doctor and you could detect whether a medicine would actually work before giving it. Which drug are you going to choose first? Anyone can answer. Okay, I'm answering first. I am going to choose paracetamol first. I hope we all know paracetamol or acetaminophen. Where I am coming from, Ghana, um, paracetamol has almost become like the first line um, solution for all diseases. So no matter the illness you or the disease you send to the hospital, you'll be prescribed paracetamol or paracetamol will be added. It's necessary sometimes, but um, it has become it has gotten out of hand. So because of this, this issue, this is what happens. Patients or people who suffer from diseases, instead of them going to the hospital for the physician to take care, to diagnose and then treat what is wrong with them, they know that when they go, they're going to be given paracetamol. So they'll just find a pharmacy or a drug shop nearby and then buy the paracetamol because they know that's what they are going to be given, and then take them and then expect that they will get better. Because, and it's true, when they go, mostly it's paracetamol. And then my auntie has been a victim of this situation. I think last year or last 2 years, she broke her bone. She had an accident and went to the hospital, and she was given 30 sachets of paracetamol tablet, and one sachet has, I think, 10 tablets, so 300 paracetamol tablets for her to finish taking within 2 to 3 months. Yes, she didn't take them, by the way, but I want you to understand how serious it can be. So if, let's say, if my auntie has a variation in the gene, the CYP3A4 family of genes that are in charge of metabolizing the paracetamol, if my auntie has a variation and is not able to metabolize the drug, she will be taken, but it's actually not able to metabolize, and it's not physical. We cannot know until she has undergone genetic testing. She will take the drugs and then hoping that she would get better or the drug would be solving her problem. But the drug would be causing serious problems. It would be attracting other problems for her, like for her to add to the bone, the broken bone that she has. So it can lead to drug toxicity, which can damage her organs, especially her liver and her kidneys, and that is very dangerous. So, if I am a medical doctor and I can actually predict, I would choose paracetamol. Yes, I would choose paracetamol because it's something that is being prescribed for everyone, so that I would know if it will actually work for this person, it will cause this effect in this person, or it's unsafe, I have to change it. Yes, so I have answered, and I'm waiting for your answers as well. So anyone, you can just raise your hand and then unmute yourself and share with us. Yes, Pascal. Pascal, the floor is all yours. Okay, okay. Come again, please. I didn't really hear you.
Oh, okay. Yeah, I think I can speak now. Can you hear me?
Yes, Pascal.
Yeah, I think I'll go for sulfonamides, especially the bacterial sulfonamides. So we have sulfonamides are used in anti-malaria like Amala, Fancida, as sulfadoxine, and then also Septrin, the popular Septrin as sulfamethoxazole. And the reason I'll go for that is because of a very popular pharmacogenomic important factor, the G6PD deficiency. So that's like an enzyme, the G6PD enzyme, that when probably due to genetic factors, one lacks that enzyme, there will be some medications that triggers hemolytic anemic symptoms, and sulfonamides fall under that. When I prescribe sulfonamides to some individuals. I've witnessed a case where we prescribed sulfonamides, particularly amala, to that individual, and it triggered a cascade of adverse reactions. And the reason was most likely, although I don't know if it went to the point of lab scrutiny or whatever, but our preceptor told us most likely it was due to G6PD deficiency in that patient, because that patient's spouse could tolerate the amala, that's the fancida medication, well. So I think I would like to, like, if I'm the one, I would want to test on sulfonamides. I would want to test on GP6D deficiency on patients to see if they are genetically predisposed to that deficiency before I prescribe any sulfonamide like medication.
That is very insightful and very interesting. That is. Thank you very much, Pascal, for sharing with us. And yeah, it can prevent like a whole lot of complications. It's very important in this regard. And please, anyone to share with us. Oh, no one. Or you don't want to be doctors. I'm still waiting.
I think we can just move to the questions. I think maybe they have more of questions than
Okay,
contribution,
okay.
as it were.
Sure, sure. So let me just quickly sum up. So we've talked about pharmacogenomics and how that leads to personalized medicine, medicine that is designed and tailored based on your genetic profile, and how that improves outcomes and saves a lot of money for hospitals and HMOs, and also why it's very important for Africa. It's actually important globally, but Africa to be precise, based on our rich genetic diversity. So thank you very much for being with me up to until this point, and thank you for your contributions, everything. Thank you, Pascal, and I'm ready for your questions. Any question that you have, I'll be glad to address them.
Okay, thank you very much for that great presentation, sir. I believe
You
we
are
have
very
all gained
welcome.
one or two. Yeah, so we're going to get to the question and answer session briefly. Please, you can signify if you have a question, or you can share the questions in the chat box.
Yes.
Okay, Mr.
You can
Pascal.
type it so that I can read the questions in the type box if you want to do that.
Okay, Mr. Pascal, you can speak.
Oh, okay. Thank you. Thank you, sir. I really appreciate it. Thank you, Mr. Paul. There are two questions I want to ask. One is a bit compound. I don't know how to put it in words, so I'm hoping you would understand what I'll be trying to ask. And the second one is pretty much straightforward. So the first one is, when it comes to pharmacogenomics research, or let's say, pharmacogenomics investigation in hospitals or clinical settings, I'm going to assume or presume that one of the goals is to reduce costs, because you talked about not wasting drug, not wasting money. Yes. Am I right about that?
Yes, yes.
Okay, but then what I thought during the lecture was, when it comes to pharmacogenomics, I don't think it's straightforward, in the sense that there are various genomic contributions to why people could have idiosyncratic reactions to medications. So you're looking at this, for example, now when you look at the cytochrome enzyme, the enzyme system, apart from the 2p19 or C2p19, we also have 3A4, we have various CYP450 enzymes, and some could be inducers, some could be inhibitors of the drugs. So what I'm saying is, since there are various ways in which someone can have a different reaction to medication, doesn't that kind of make the genetic investigation, doesn't that make it too cumbersome? Won't that fall back on the cost of the patient? For example, if you want to go through the pharmacogenomic route for me as a person, and you have to test for GPCD deficiency, you have to check for CP3A4 inhibition, you have to check for CP2C19 inhibition or metabolizer, like, won't that lead to me paying for a lot of tests? Doesn't the how will I put it now, the complicated ways in which one can react differently to drug, doesn't that reflect in the cost of the therapy? That's my first question. My second question is, you said something about reproducibility of or reusability of the results. So let's say they run a pharmacogenomic profile investigation of me when when I'm 20 as an antihypertensive patient, and then when I'm 40, they can make use of that. What if the contribution to that my idiosyncratic reaction is not genomic but transcriptomic in nature? I know transcriptome is not static, it's flexible. So my transcriptome profile at 30 could be entirely different from my transcriptomic profile at 50. Won't they need to then conduct another set of tests? So that's my second question.
Okay, Pascal, thank you for your question. For your first question, you are right. You are very right. Pharmacogenomics is actually expensive. It is expensive and it requires money. But the sacrifice that we will make in terms of finance, the short term would actually help in the long run. It will be more than the long term. It would make sense. It would make sense and then help us than how the long term of we not integrating pharmacogenomics into our healthcare would cost us. So it's expensive, but if we are able to integrate it and then apply it, it would actually save money because drug wastage causes us a lot of money that when we sit back and look at it, testing, doing the pharmacogenomics test, and then saving the money, like helping to tailor treatments, and then saving all the money from the risk that would have occurred. It would compensate, and even it would be more, way more than we not um integrating it and then continuing with drug wastage and all that. So I hope that that answers your question. It's a sacrifice we have to make, because in the long run it's going to the starting would be expensive and it would require money, but it's for the good. And then your second question, yes, it's true that um um when it's in terms of transcriptome, it can be flexible. But it's a really interesting question. It's something that I would look into, and then right now I don't have solid explanation or solid response in that regard, but I would look into it, and then maybe we can discuss about it later or in the future. But it's an interesting question. Thank you very much for your questions, Pascal. And I hope I have answered the first one in a way.
Yeah, yes, you have. Thank you very much.
You're welcome, Pascal.
Okay, I also have a question. Can you hear me?
Yes, please. I can hear
Okay.
you.
Okay. Thank you very much, Mr. Paul, for the session. We really appreciate you. So my question is, I'm not sure if maybe you captured it in the session, maybe at the beginning, or but I just felt like I should ask because I've not gotten the answer. So I'm trying to ask that is, let me say this, like, is pharmacogenomics like really worth exploring? I'm going to ask this question by making an analogy. Okay, so I did biochemistry in my undergrad, and I remember very well that there were we did a lot of abnormality studies as it well. So we'll do a pathway, let's say we do glycolysis, let's say we do carbohydrate metabolism, and we're so it's okay, pathway glycolysis. So after all that session, like after that class, there's always a part where we um, study the different diseases, conditions that could occur as a result of like any aberration in that particular pathway. So those conditions are always very, very vague. And at the end of it, what we hear is that it occurs in only maybe 1.5% of the world population. It occurs in only 0.5 of the world population. So that thing always like, I don't know, but I always felt like it's a waste of time because it does not have a global relevance, because it affects very few people, like very, very few people. You can understand what 0.5 or 0.01 of the whole world population equals to. So my question now is, if we're talking about pharmacogenomics and we're talking about variation in genomics, so how are we sure that the variations in genomics are going to be explored in pharmacogenomics and not also, for example, you were mentioning some during the two scenarios you mentioned some, what's it called, some enzymes. You said, okay, this person has this particular variation of this enzyme that does not allow them to do this. For those enzymes, what are the statistics? Like, can we say approximately 50% of people, like how many, what's the probability of somebody having that variation in their gene that that will make the journey of actually exploring that variation worthwhile? I don't know if you understand my question.
Yes, I think I do. So um where do I even start? So yes, the global relevance aspect, 0 point, um like you think that that percentage is irrelevant, and then it doesn't really it's not of deserving committing so much by integrating pharmacogenomics, the cost and all that. Yes, but I believe that with time, due to newer technologies, AI, pharmacogenomic testing is going to the cost is going to reduce, because it was way expensive like in the early 2000s. But if you see the statistics, the cost has reduced drastically till now. Today, genetic testing is not as expensive as it was, due to the new and then like the simpler and affordable technologies that are coming into play. So I think it's expensive. It's a sacrifice that we have to make for the better good. So with time, I believe it's going to reduce, and then these healthcare facilities can collaborate with research institutions and all that, and this will further reduce the cost involved. And then your second concern, um, I I I didn't really get that aspect. If you can simplify it for me.
It's just one question. I was just asking, like, where's the relevance, like the global relevance, like those variations, what's the probability of somebody having, let's say, this particular there is a, I don't remember the first scenario, I don't remember the name of the enzyme, about the what's it called,
Oh, the
the
codeine.
boy
The
with,
codeine.
yes,
Okay,
yeah, the
okay.
boy that took codeine and easily metabolized into morphine and went into coma. That particular enzyme that actually led to that was a to that condition now.
Oh,
It
it's
was the
actually
current, like the what's it called, the statistics. What's the probability of somebody having that what's it called, that enzyme, which
it's
will make them prone to that condition?
it's actually that's because I the reason why I use that scenario, those two scenarios, is how common they are. Codeine metabolism variations is very common in Africa, and it causes a lot of death. Yes, there are a lot of research papers around that. That is why I specifically included that. So it is quite common. Codeine and clopidogrel. Yes, it is quite common. So if pharmacogenomics comes into the picture, it is really going to help. It is going to really help and transform healthcare.
Okay, thank you very much. Actually, your answer has also brought up another question, but I won't
Okay.
want us to what's it called, exceed. We have actually exceeded our time frame. So maybe I can reach out to you on LinkedIn and ask my new question that I just thought of as you answered. So thank you very much for the session. Ms. Gladys, you can go ahead.
Okay, I was going to say I have a slide. I have a slide of the references, the research papers that I got my information from, and that proves how common these metabolic variations are, especially in the African population. But it's not here, unfortunately. So I would adjust the slide. I will send you that list of references so that it can help answer your questions.
Okay, thank you very much. I think that will be fine.
You are very welcome. Please, any other concern?
Please, do we have another question? Okay. So in the absence of no question, I'm going to invite our I'm going to invite our team lead to brief us. Uh, just a moment now.
Okay, thank you very much. Pastor, I can see your hands up. Do you have anything to say?
Oh, okay, it wasn't a question. It was something like I feel like I just wanted to say I think one way for pharmacogenomics to really move far in a place like Africa is to combine it with something called pharmacovigilance. So basically, pharmacovigilance is the reporting of reporting or observation of the adverse effects of drugs when they are now in circulation. You know, when during testing of drug, the phase, the clinical trials is not tested across all demographics. Pregnant women are not tested, children are not tested most times. So when it's in circulation and is available to all and sundry, there is still a sort of pharmacovigilance where there is an investigation on or observation on what the drug does. So I think if there is more reports on pharmacovigilance observations, it could channel pharmacogenomic research, because when we see that, oh, this drug is affecting a particular set of people, or this drug tends to have this adverse effect around this particular time, it really directs pharmacogenomics investigations better. But in Nigeria, with poor pharmacovigilance reports, it doesn't help the pharmacogenomic case. So that's what I wanted to say.
Okay, thank you very much, Pascal,
Thank
for
you.
the
Thank you.
contribution.
It's a really insightful one because integrating pharmacogenomics and concentrating on pharmacogenomics alone would be sort of a challenge. So it would actually help when we support it with other disciplines like you've mentioned. Thank you, Pascal.
Okay, so we've come to the end of the session. Thank you very much, Mr. Paul, for joining us this evening. We really appreciate your presence, and I must confess it was an awesome, insightful, and enlightening presentation from you. Thank you very much. Okay, so I am for the participants. I am Mutumare Lesomi. I am the team lead for Genomax Services and Consults. This webinar was sponsored by Genomax Services and Consults. Genomax Services and Consults is a subsidiary of Genomax Holdings. If you are conversant with LinkedIn, you must have seen our flyer or name somewhere. So we are a bioinformatics servicing and consulting firm. We offer bioinformatics services to institutions, to researchers, to hospitals, and the likes. We also offer consultations to researchers. So maybe you are a PhD student, an MSc student, and you're willing to do your project in bioinformatics. You want to apply bioinformatics to your project, but you're not sure how. You have not been able to design the methodology. You feel like you don't even have the skills needed. You can reach out to us. Yes, you can book a consultation session with us. We'll work with you to design your project and even train you. Yeah, we do personalized training too. So we can work with you, mentor you, and train you through your project to the end. So thank you very much, Mr. Paul, and we really appreciate everybody for joining in. We are going to be having another session come next month, which is in January 2026. So please be on the lookout for the flyer, for the details. And the link for the attendance has been shared in the chat box. Please don't leave. Please make sure you fill the link for the attendance. Fill your name as you want it on your certificate of attendance. So it's going to be used, that link, that what's it called, that data is going to be used to generate the certificate, and you will have your certificate very soon. So thank you very much, and do have a pleasant evening. If you have not filled the link, just stay on the call to fill it and you can leave. Thank you very much. Thank you, Mr. Paul. Thank you thank you, everyone.
You're welcome, and thank you too. Thank you very much for having me. Thank you.
You're welcome.
Thank you.
Yeah,
I
thank